Hi guys, welcome to class. I want us to consider the anatomy of a simple bacterial cell. Now, a bacterial cell is also regarded or referred to as bacterium. There you go. Now, this is just the anatomy of a simple. This diagram is showing us the anatomy of a simple bacterium or a bacterial cell. Okay, so what do we got? Now, we're looking at a prokaryote. Don't forget, a bacterial cell is a prokaryotic cell, in the sense that you can't see any well-defined nucleus. You understand? And that's very evident. Everything is just floating within the cytoplasm. There's no well-defined nucleus. There are not also compartmentalized structure within the cell, which we call organelles. Everything is just free for all, just floating and floating and floating. Now, basically, we're looking at, now what are the things you can identify? Now, first of all, you need to know that it has a cell wall. Now, which other common cell do we know that have a cell wall? Or how should I put it? Well, let me just go straight to the point. You know, that's what differentiates your plant cell from your animal cell. Your plant cell has a cellulose cell wall. Your animal cell doesn't have. But in the case of the bacteria, it has a cell wall, but the cell wall has a different constituent. What do I mean? This is what I mean, actually. So, anatomy of a bacterial cell. Anatomy of a bacterial cell. Bracket bacterium. Okay. Now, we have first identified the structure, cell wall. That's what we just identified, right? Now, what is the unique thing about the cell wall? It is made up of a peptidoglycan matrix called murine. It's made up of a peptidoglycan matrix. Made up of a peptidoglycan matrix called murine. So if you ask what is the composition of the cell wall of a bacteria, the solid perfect answer will be what? Murine. Bless you. Now, murine. Okay, so let's get back to our diagram. Yeah. So I've identified the cell wall. A murine, a cell wall composed of murine. Let's not destroy English. Now, what else can we see? Yeah, of course, in addition to that, it also has a cell membrane. Or sometimes it can be referred to as a cytoplasmic. Most times you can also call it a cytoplasmic membrane, plasmic membrane. Now you can see that there's this region in the bacteria which they call nuclear region. And that's where you see the nuclear material. The nucleus. Yeah. Now, also, the bacteria has extra chromosomes, extra chromosomal DNA. Now, the unique thing about those extra chromosomal DNA is that they actually, when the bacteria is dividing and is transferring its genetic material, most times, in some cases, the plasmid doesn't go along. So, basically, the plasmid doesn't really have any function per se. But scientists have discovered that these plasmid, which are extra chromosomal DNA, so plasmid, we will say is a chromosome. We can describe it as a chromosome contained in the cytoplasm of a bacteria, contained in the cytoplasm of bacteria, stop. It carries extra genes. It carries extra genes. Or you can say is an extra chromosomal DNA. So now this plasmid, scientists do say, though it doesn't have any specific function, it just carries extra DNA. But it is found that it confers special ability on the bacteria. It is found that it confers special ability on the bacteria it confers special ability on the bacteria, which includes resistance to antimicrobial treatments, which include resistance to antibacterial treatments. Of course, when you come down with a bacterial infection, let's say stomach flu or gastroenteritis, you want to take antibiotics. Now, Nigerians abuse antibiotics a lot. So if you are one of them, please do stop that. Because when you abuse antibiotics, you are just, that's the reason why you finish 2, 3, 4 packs of antibiotics and you are not still cured. Then you then resort to herbal treatment, and after a day or two you feel okay. Then you say white man doesn't know what he's doing, white drugs don't work. It's you that didn't make the drugs work. Because when you use antibiotics too much, you are just destroying the normal flora of your body, predisposing you to more infections. And that is why you should stop abusing antibiotics. When you feel is a bacterial infection, you use an antibiotics, you do not see any change, you go consult your doctor. That is how you should use antibiotics. It is found that it confers special ability on the bacteria, which includes resistance to antibacterial treatment. Yeah, so that's what they found out about plasmid. So things like resistance and also not only resistance to antimicrobial treatment, ability to resist some very harsh conditions. Scientists have found out that those things are related to the plasmid. So it's the plasmid that gives them those extra features, basically. So these are organelles. So in most cases, in most bacteria, you won't see organelles because you just see those structures just lying freely. Cytoplasm. Of course, you have ribosomes, which of course is for protein synthesis. And your bacteria has a 50S and a 30S ribosomal subunits. Do you understand? So you have the 50, you have the 30. So the heavy is the 50S subunit, while the lighter is the 30S subunit. And most times, most antibacterials like your tetracyclines, which you commonly use, they will inhibit protein synthesis at the 30S subunit. Of course, you know ribosome is an organelle that functions for protein synthesis. Don't forget your cell theory. Now you then have a very important structure which you call pili. That's for plural, pilus for singular. And this structure extends from the cell and it functions for transferring genetic material into another cell, particularly during the process which we refer to as conjugation, which is another way of reproduction which the bacteria undergoes. And of course we have the flagellum, Vibrio cholera. Now you have it present in this species of bacteria we call Vibrio cholera. In some cases you could have two, so you just have a single, we say flagellum, and if it's double like a pair, we say flagella. And of course, what's the function? It's for movement, locomotion. So this is just like an overview, a simple anatomy of a bacteria cell. So see you in the next class.